Hi everyone, I'm Abigail Williams. I'm a freshman student at the University of Charleston, and I'm a biology major with a concentration in environmental biology. This presentation is on a research project that I was part of last fall semester, and we researched hiking trail use impact on plant community composition and soil chemistry. So this is the area that we studied. It's Canal State Forest in Charleston, West Virginia. Here are the hiking trails that we were studying. You can see here is Poly Hollow Trail, up here is White Hollow Trail. All of these little drop points are our sampling locations, and White Hollow Trail was a single-use hiking trail, and Poly Hollow Trail was multiple-use hiking trail where there were also other activities. So the problem that we were looking into is habitat loss and fragmentation, and it is a major environmental problem for our forest, and it is a major cause for biodiversity loss. The West Virginia State Wildlife Action Plan listed in 2015 that the top stresses on our terrestrial and aquatic habitats were invasive non-native species, roads and railroads, utility service lines, housing and urban areas, and recreational activities. As we build our hiking and riding trails in our forest, we must consider the effect that their presence and use will have in the plant communities and in the soil quality of the forest. So to introduce our research process, the objective of the research was to determine if the hiking trail use impacts the plant community composition and soil chemistry. And the hypotheses that we had about this study were that the effects of the trails will be more prevalent on the multiple use trail than on the single use trail, and that the type of trail use will have an impact on the plant community composition and introduce more invasive species, and that the type of trail use will have an impact on soil chemistry and quality. So the methods that we used to get our plant data were that we placed the quadrats, like in the picture, in random locations every 20 meters along the edge of the trails for 100 meters. And then we also collected samples every 20 meters, 5 meters away from the edge of the trails. We also did a stem count and identified the plant species within each quadrant using the Seek by iNaturalist app. And then we took all of our data and put it into a vegetation matrix and analyzed the data using JASP computer software. And for the soil methods, we collected our soil samples from the topsoil at each quadrant, where we also took our vegetation data along the trails. So our different variables for the soil were soil moisture, which we measured using the gravimetric method, where we dried out the soil in an oven to measure the moisture content. For the pH, we used a vernier probe in a soil slurry. And then for the nitrogen and phosphorus content, we used a soil slurry and Less Relief 1601 rapid test, nitrate, and phosphorus test containers. The results that we got from our plant study, we measured using Shannon's diversity index, and we also looked at the invasive species that we found. So for the single-use trail, the diversity was higher away from the trail, and for the multi-use trail, it was higher near the trail. And this is due to the invasive species that we found. As you can see, away from the single-use trail, we found invasive honeysuckle, and near the multi-use trail, we found Japanese stiltgrass. And the presence of these invasive species is what influenced the higher diversity index, because there were more of those individuals than there were compared to the native species, and there were so many of them present. For the soil results, we have these charts. They show through the darkness of the color, the higher the value. And for the soil moisture variable, near and away from each trail, there was not a very big difference, and there wasn't a very big difference between the type of trail either. And the same goes for the soil pH. There was no big difference near or away from the trail on either one, or between the two types of trails. For the soil nitrogen content, there was also not a big difference near or away from each trail, and there was also not a big difference between each type of trail. But for the phosphorus, it was actually slightly higher near the single-use trail than away from it, and there was not a very big difference near or away on the multi-use trail. But the slightly higher near the trail was the only big difference for the soil phosphorus. So to conclude the findings, invasive species were found in both trails, and the Japanese stiltgrass was more abundant on the multiple use trail. Invasive honeysuckle was more abundant on the single use trail. And the soil chemical variables that we measured didn't correlate with the plant diversity changes. So there's a possibility that other soil variables that we didn't measure in this study could influence those changes that we saw in the plant diversity. So this leads into our ideas for future research that we could look into. We could study seasonal changes in different locations rather than just during the same time period and looking at the same trails in the same area. We also look into different soil biological variables that might give us more sight into how it affects the plant life too. And if we want to look into the invasive species more, we could also look into those in their own individual study where we actually count those individuals rather than random sampling like we had in this, because we actually saw a lot more individuals of those invasive species along the trails, but they just weren't noted in our data because we did the random sampling. So it just didn't capture them in those quadrants. So it didn't give the full picture of invasive species on those trails. And here are my references. And thank you all for coming. And if you have any questions, I'm here to answer.